Hello, my name is Vikram Mulligan. I co-head the Biomolecular Design Group in the Center for Computational Biology at the Flatiron Institute. And today I'm going to tell you about pipelines for computational design of peptide macrocycle therapeutics that my team has developed and which can inform the wet lab experiments that are involved in discovering a new peptide therapeutic. Many of us, of course, are interested in drug discovery, which is currently a very inefficient process, as we all know. The drug discovery pipeline typically starts with screening large libraries of hundreds of thousands of compounds, with the hope that somewhere in all those compounds there are a few molecules that have a desired biological activity, which I'm showing in cyan here. As you move through high throughput screens in plates in the lab, to low throughput screens, to animal studies, and ultimately to phased clinical trials, you weed out more and more possibilities and spend more and more effort on the remaining possibilities. Nevertheless, even when you get to phase 3 clinical trials, the majority of drugs fail, which is hugely inefficient, not just in terms of money, but in terms of irreplaceable resources like your pool of patients that you can test a drug on. So we want to make the drug design process more efficient by trying to ensure that the initial pool of designs is much enriched for actual hits, and further enriching the pool by screening out as many of the misses as possible by computational means even before we get to experiments, so that by the time we get to experiments we're working with a lot of really good molecules that have a very high chance of making their way through clinical trials. That's the hope anyway. And of course we're interested in particularly doing this for peptide macrocycle therapeutics. Now, what do we mean by design? Well, peptides and proteins are long chains of amino acids. With natural proteins, most proteins fold into intricate three-dimensional shapes that give rise to their functions. And we want to produce new three-dimensional shapes that have new functions. So the challenge is, can we come up with a new sequence that gives rise to a new fold that is functional with a new desired function? Now, we can use generative AI for this a little bit if we want to restrict ourselves just to the canonical amino acids. But generative AI has trouble extrapolating beyond what it was trained on. And if it was trained on natural proteins, it's going to have a really, really hard time modeling a non-canonical amino acid. Chemists have come up with thousands of building blocks that we can and should be using to develop drugs, and we want to use those, so we need general physics-based methods. There's a big problem with peptide therapeutics, though, and it's this. If you have a small molecule and it binds to some target of therapeutic interest, shown in green, we have the problem of off-target effects. So that small molecule is probably going to present fairly similar interactions to bind to a bunch of other molecules, a bunch of other proteins, shown in blue, and give you those side effects. And so the rationale with peptides, of course, is we make the molecule bigger, we have more favorable interactions with the target, so that we're a little more selective for the target. The trouble is, as the molecule gets bigger, it gets floppier. And if it's flexible and floppy in solution, and it has to order itself in order to bind, there's an entropic cost associated with binding. And that pulls the equilibrium towards the unbound state and reduces affinity. So you lose affinity as you make the molecule big and flexible. In addition, if the molecule is too flexible, maybe it can flop around into an alternative conformation where it could bind to an off-target protein and produce side effects, which is what we wanted to avoid in the first place. So the solution is make the molecule rigid. If we can design peptides to fold rigidly into their binding competent conformations, then they can't flop around into alternative conformations to bind to something else, and there's very little entropic cost associated with binding. The molecule is pre-organized in its binding competent conformation and can bind with high affinity. In order to do this, we use a set of tools, the first of which is the Rosetta software suite. Rosetta is old software. It's been the state-of-the-art for physics-based protein modeling for a long time, developed in the 1990s and gone through many iterations of rewrites. And at its core, it's a set of tools for keeping track of long chains of amino acids and treating them as kinematic chains that you can manipulate in various ways, putting them in different conformations or designing different sequences. And on top of that is a physics engine for computing energies. And on top of that are all sorts of protocols for particular tasks: docking, design, simulating folding, etc. Now, we've developed in Rosetta a pipeline for designing rigidly folded peptide drugs. This pipeline consists of starting with a structure, typically a high-resolution crystal structure, of a target protein, sampling many, many backbone conformations of peptides in some binding pocket on that protein. And for every plausible conformation that could be stabilized by a suitable choice of sequence, we apply fixed backbone sequence design algorithms, and we make thousands and thousands of designs. And then we throw away as many of them as we can. We apply low-cost computational filters to identify features that are obviously incompatible with success. And once we have a shorter list that pass filters, we perform high-cost simulations, particularly folding simulations that I'll talk about in a moment. And this allows us to rank the remaining designs to prioritize them for the highest cost thing of all, which is synthesis and wet lab experiments. As we go from left to right here, we go from many candidates to few candidates, and we go from very few resources spent per candidate to lots of computational and ultimately wet lab resources spent per candidate. The idea is to fail fast, succeed slowly. Now, using this pipeline, we've made many designs over the years. These are all molecules that we've designed on the computer, synthesized, and solved either X-ray crystal or NMR structures of. And here I'm showing the experimentally determined structures. And these peptides are made from mixtures of L and D amino acids. And they can also incorporate other exotic chemical building blocks, like the three-way crosslinker that you see in the middle of the molecule on the right. They also have exotic structural features that you don't see in natural proteins, like the helices of opposite-handedness that you see in peptide C here. Now, these ones were just designed to fold into pretty structures, but we've also made functional peptides, such as this guy here, which binds a central zinc atom and buries polar groups, shown in cyan, and exposes hydrophobic groups, shown in orange, forming kind of an inside-out fold. When you chelate away the zinc, the peptide flips around inside out, so it acts as a conformational switch that's sensitive to zinc. And in the absence of zinc, it buries its hydrophobic groups and exposes its polar groups like most proteins. This could be one day the basis for designing membrane-permeable peptides, peptides that can adopt a conformation in which they expose hydrophobic groups to pass through a lipid bilayer and then expose polar groups to be soluble in the cytoplasm again. And of course, what we're interested in ultimately is designing peptide therapeutics. And we've had some success with this. This is a paper that we published a few years ago in PNAS showing a designed inhibitor of the New Delhi metallo-beta lactamase 1, NDM1. NDM1 is an antibiotic resistance factor, so if we can inhibit it, we could make all the old antibiotics useful again. The peptides that we designed, and this is one of them, folded into structures very, very close to the design models. So in light colors here, I'm showing the model that came out of the computer. In dark colors, I'm showing the X-ray crystal structure. And you can see that there's almost a perfect overlay here. We can control the arrangement of matter with sub-angstrom precision. And that's particularly significant because this is a molecule made of a mixture of left and right-handed amino acids, natural and non-natural amino acids. So we couldn't have done this with generative AI. We had to use physics-based methods. Most significantly, this molecule binds where it's supposed to in the orientation it's supposed to. So in green, I'm showing the target protein, NDM1. In pink and cyan and orange, I'm showing the peptide. And on the left is the computational model, the fiction that comes out of the computer. On the right is the actual experimental reality. Arginine 1 is not visible in the crystal structure, but aside from that, you can see that the peptide is binding in the desired binding orientation, the desired binding mode, at the desired binding site, all of which is stuff that you don't get automatically out of blind screening approaches. So we were successful. But most importantly, this illustrated the success of the simulations and of our working hypothesis about conformational rigidity being the key to success. And I'll go into that in a little more detail. So the last step of our pipeline before we hand it off to experiments is this high-cost conformational sampling. The question we're asking here is, for a given sequence that we've designed, does it fold into a unique low-energy state that matches the binding competent conformation, or are there alternative conformations it could fold into? To do this, we take a sequence, we fix the sequence, and we put it into zillions of different conformations, relax each one, and compute the energy of each one. And in the plot on the bottom, each of those blue points is a different sampled conformation. On the horizontal axis, you can see the RMSD to the designed conformation, the binding competent conformation. And on the vertical axis, you can see the computed energy. And we want to see something like this, where we have a nice funnel-like shape going down to a unique low-energy state that very closely matches the binding competent state. And there are metrics that we can compute that quantify the goodness of this funnel. So we use this to rank our designs. From this computation, we can also compute an estimate of the delta G of folding, the folding free energy. So here is a plot where on the horizontal axis, we're showing that computed folding free energy from that simulation. Lower is better. And on the vertical axis, we're showing the experimentally measured IC50. Lower is better. And this is for 7 peptides that we characterized computationally, and then we synthesized and we characterized experimentally. And you can see there's an excellent correlation between the extent to which the peptide is predicted to fold into the binding competent conformation, shown on the horizontal axis, and the extent to which it actually binds to and inhibits the target, shown on the vertical axis. So the correlation is so good, in fact, that we were able to predict the rank order of these peptides, which is great. There's a calculation you can do on a computer that predicts success in experiments, and that means you can reduce the number of experiments you need to do to find a hit. Now, I won't pretend that all of our computations work equally well. Intuitively, the binding affinity should be dependent not only on the propensity of the peptide to favor the binding competent conformation, but also the interaction energy between the peptide and the target. And when we compute that for these 7 peptides, we see there's no correlation with success. Now, there is a reason for this. We selected these peptides based on computed affinity for the target. So these were the best peptides out of a pool of thousands. If we were to plot the thousands, and if we were to do experiments on all 1,000 candidates, probably there would be some correlation. But among the very best peptides, we're down in the weeds where the noise dominates and we can't actually discern which are the best peptides and which are not. So there's room for improvement. The next, before I talk about how we're trying to improve this, I'll just mention one other thing. We're also able to design membrane-permeable peptides. And this was a paper we had in Cell a couple of years ago, where we designed several peptides that were able to penetrate the cell membrane by adopting conformations where they internally satisfy all their hydrogen bond donors and acceptors so that there's very little desolvation penalty associated with passing into the bilayer and losing the interactions with water. And you can see that on both PAMPA and CACO2 permeability assays, we had excellent permeability for some of these peptides. The current challenge is designing peptides that are both permeable and which bind to a target. And actually, I have a collaborator who's using these methods who has achieved that now. So hopefully we'll be publishing that soon. Now, we want to make these methods better, as I said. And there are kind of 3 areas in which we want to do that. First is accuracy. The more accurate we can make our predictions, the fewer experiments we need to perform in order to find a hit. If our success rate out of the computer is 1 in 10, we need to do on average 10 experiments. If our success rate were 1 in 2, we would on average need to do 2 experiments. Second, we want improved generality. The chemists keep adding new chemical building blocks that we can work with, and we have to keep doing a lot of work in order to be able to use those chemical building blocks with our force fields. So we want to make it easy for biologists and chemists to try out new scientific ideas and experiment with new approaches. And we want to improve the tractability and efficiency for design. The more efficiently we can do our computations, the more possibilities we can consider on the computer, and the less we have to do at the bench. So one of the ways we're doing this is with the new approach that we've developed called macromolecule QM. Proteins, as we usually model them, are little balls that exert forces on one another, and we make this Newtonian approximation. But a better model of a protein or a peptide is that it's a bunch of atomic nuclei surrounded by a cloud of electrons, where the electrons are described by the Schrödinger equation. The quantum chemists have come up with better and better approximations for solving the Schrödinger equation in order to describe this cloud of electrons and compute properties from it, like energies. And some of these approximations scale very well to systems the size of peptides and even proteins. We're not using them very much in our modeling pipelines, though. So macromolecule QM is a set of tools that allow you to run whatever QM calculation you would like in the context of whatever peptide or protein design or validation pipeline you like. And this leads to greater accuracy, but also greater generality, because QM calculations are totally general. As a demonstration of that, we've applied this to computing the Ramachandran map for amino acids. The Ramachandran map is a plot of the conformational preferences of a given amino acid. And it's a two-dimensional plot as a function of two dihedral angles, phi and psi. These are the main chain torsion angles in an alpha amino acid. If you look at all the amino acids in all the proteins in the protein data bank, you find that these phi and psi values are not distributed randomly. Rather, they have a clear distribution to them, where certain regions of Ramachandran space are favored, particularly those that give rise to alpha helices or beta sheets. And in Rosetta, we use a force field that has a Ramachandran potential as one of its terms. This potential is taken from statistics from the protein data bank, and it looks like this red and blue plot here. Now this potential, because it's based on statistics from the protein data bank, misses some features. We get less accurate in the high energy regions and in the slightly strained conformations. And some of the nuanced little jiggles that should be there aren't there. By quantum chemistry methods, we can compute much more accurate and precise Ramachandran potentials. And these are shown below here. So this is the potential for alanine. We've done this for all of the 20 canonical amino acids. Leftmost column here is the structure of the amino acid. Then the next column is the statistical potential, and the column after that is the QM computed potential. And you can see that the QM potentials are similar, but they have some different features. Now, why do we think that these are better? Well, one answer is if we take the alpha helix propensities computed by QM, shown on the vertical axis here, and we compare them to experimentally measured alpha helical propensities for each of the amino acids, we see a pretty good correlation. And we were glad to see this because this means that we have some confidence that this gives us generality as well. QM calculations have no parameters that have to be calibrated specifically for an amino acid type, so we can apply this just as easily to exotic chemical building blocks like AIB, or to even more exotic chemical building blocks like peptoid monomers. Peptoids are like amino acids, but the side chain comes off the nitrogen. So here's a chiral peptoid where we've computed the Ramachandran potential. And this allows us to extend our force fields to allow much more exotic chemical building blocks to be modeled. In addition to building better force fields, we can use macromolecule QM-based calculations in any of our existing pipelines. So one example is we've extended the structure prediction and conformational sampling pipeline that I showed you earlier to incorporate a QM-based geometry optimization step and then a QM-based energy calculation. And we can do this in the context of massive computing clusters where in parallel we're sampling zillions of conformations across many different CPUs on the cluster, and we're performing many QM calculations in parallel. So doing this, here's the old result using Rosetta's force field, where we're trying to predict the structure of cyclosporin. Cyclosporin's a great test case because it's a cyclic peptide with exotic chemical building blocks, a D amino acid, lots of N-methylated amino acids, a couple of weird non-canonical side chains. It has all the features we want to incorporate into drugs that we design. Rosetta's force field does very poorly with it. It predicts a mess. So in green, I'm showing the X-ray crystal structure of cyclosporin. In magenta, I'm showing Rosetta's prediction, and it just gets it wrong. But when we use a QM calculation, and here every point in the plot on left is computed using QM methods on a massively parallel supercomputer, now we get it totally right. In green is the X-ray crystal structure. In cyan is the prediction that comes out of Rosetta predictions using QM calculations. And you can see that we have sub-angstrom precision, not only predicting the backbone conformation of cyclosporin, but even the side chain conformations. Not only that, we predict accurately the cis or trans isomerism of all of the peptide bonds. So this is great. Now we've got a pipeline that we can be even more confident of for predicting the structures and validating the folds of peptide macrocycles that we design. Many of our tools we're building into a new software suite called Masala. Rosetta, unfortunately, is not open source. Masala is. And ultimately, we want Masala to be a successor to Rosetta. Masala is also an opportunity to rewrite a lot of our algorithms to take better advantage of modern massively parallel CPU and GPU hardware. And it's got a plug-in architecture that makes it easy to extend it. It's easy for a biologist to extend it. It's intended to be used as standalone software, but also it's intended to be used as a support library for other software. And, among other things, it can plug into Rosetta and extend Rosetta's functionality. One thing that we've done is revisit the sequence design algorithm. We've made it much faster on CPU. Here is a plot of Masala performance versus Rosetta performance for a design task, for a bunch of design tasks. And you can see we can get up to 2 orders of magnitude of speed up just on CPU alone. We also can parallelize this. This is not even parallelized. Rosetta was written for an era in which computers had one CPU with one core and would do a series of things in sequence. So that peptide design and validation pipeline that I showed you, we sample backbones on a single CPU core, and then for each backbone conformation that we have sampled on a core, we do some sequence design. The sequence design is done for one sampled conformation. We finish that, we say that's our design, and we go on to the next one. And if we want to parallelize it, we use many CPU cores on a big cluster where each one is doing an independent job. This isn't very efficient. Modern CPUs operate much better on vectors of inputs, producing vectors of outputs, and GPUs especially really like large vectors of inputs, producing large vectors of outputs. So we're trying to update our pipeline to take advantage of that sort of hardware. And so we've got the conformational sampling working now on GPU, producing many samples all at once. We've got the sequence design working on GPU, taking many input backbones, producing many output sequences all at once. We're just putting the whole pipeline together now. The hope now is that even on a single node on a cluster, you'll be able to do many designs at a time rather than one at a time. And if you parallelize this across many nodes of a cluster, you'll be able to do many at a time. So this improves our efficiency and allows us to consider many more possibilities. I have 2 postdocs who are working on this. Xiao Zhu has developed the backbone conformational sampling, and Barjeen Turzo has developed the sidechain sampling and the sequence design algorithms on GPU. Both of these are excellent, capable postdocs who will be looking for positions soon in case you know of any positions that are available. Now let's take a closer look at the sequence design problem and how we solve it currently and how we want to solve it in the near future. And this is the last part that I'll talk about here. Sequence design is the problem of given a backbone conformation that has already been sampled, like this gray backbone here, and given a number of positions on the backbone where you want to assign the sequence identity, and given a bunch of candidates for the sidechain conformation and identity, so not just the sequence identity, but the different conformations for each possible amino acid identity you could assign, we want to choose one sidechain conformation and identity per position such that a function is minimized, usually an energy function. So this is the fixed backbone sequence design problem, or rotamer optimization problem. And it is unfortunately a hard problem. You could try it by exhaustive enumeration, but the number of possibilities, the number of combinatorial possibilities, rapidly grows intractable as the number of positions or the number of possible building blocks grows. You can do it by branch and bound methods, where you reject branches of the decision tree. This makes slightly larger design problems tractable, but it still rapidly grows intractable. So what we found works best is Monte Carlo methods, where we do kind of a drunkard's walk through all the possibilities, and pretty quickly we find a good possibility, but we sacrifice any strong guarantee of finding the very best possibility. Once we've found a rotamer assignment, a set of candidate sidechain identities and conformations, we map that back onto the structure and we say, okay, that's the best we've seen. That is our designed structure with a designed sequence. And because we're assigning both the sidechain conformation and the identity, we've assigned the identity, which means we've assigned the sequence. Great. This is how we're doing it right now. This works for problems up to a certain size, but we have d rotamers per position and n positions, which means that we're searching a space with d to the power of n possible solutions. Now, the chemists keep making d larger and larger. They keep coming up with new chemical building blocks we could use. And of course, we want to make larger peptides and larger proteins, which means that n gets large. And that means d to the power of n gets very large very fast and becomes large enough that no known algorithm will solve this problem efficiently. So we need to be thinking about the future as well, and thinking about, are there better ways we could be solving this problem? And to that end, we're starting to look at quantum computing. Quantum computing is an idea that was introduced in the early 1980s by Richard Feynman. The idea was use quantum physics to solve very hard problems that classical computers just can't do well. On classical computers, to solve a difficult optimization problem, you use some form of iteration, where you assign scores to each of your possibilities, and you consider a bunch of possibilities, and you pick the best one that you consider. You can parallelize this, you can use branch and bound methods or whatever else to make this more efficient, but ultimately at the end of the day you're going to be considering many possibilities and one at a time. And that limits how many possibilities you can consider. On a quantum computer, quantum computers instead of bits have qubits. And if you have a qubit that can exist in two states, and you have q of them, you have a computer that can exist in 2 to the power of q states simultaneously. So if you assign a different solution to your optimization problem to every possible bit string, you can consider all of them at once. Now that's fine, you can consider all of them at once, but how does that help you? Well, by applying a suitable quantum algorithm, you can enrich the best one and deplete all the others, so that when you measure the state of your computer, that's the one that you're overwhelmingly likely to see. So this is a totally different way of solving these really hard optimization problems. Sounds great in theory. Can we make it work in practice? Well, the answer is yes. We mapped the problem to the D-Wave 2000Q quantum annealer, which was an earlier generation of quantum annealer. And on it, we designed peptide heterochiral helical bundles made from alternating D peptides and L peptides that were designed to self-assemble into this three-dimensional structure. We made a bunch of designs. We synthesized 2 of them. We got an X-ray crystal structure of 1 of them. In cyan, I'm showing the model that came out of the quantum computer. In orange, I'm showing the X-ray crystal structure. And you can see that we very successfully designed this molecule and designed the interface between these helices. So this works. There's a caveat here. This wasn't better than a classical computer. We could have solved this problem on a classical computer. It was a non-trivial problem, though. We couldn't have solved it by human intuition or by pen and paper or anything like that. It required a computer. Now we're trying to make this better, and we've found approximation methods that allow us to compress the problem and scale up to even larger design problems. So now we're trying it with designing entire proteins on the quantum annealer. And we're getting very plausible designs out that have nice, well-packed apolar cores, shown in orange, and nice polar surfaces, shown in cyan. Because we're trying it right now with canonical amino acids, we can put these designs through AlphaFold to validate them. And AlphaFold predicts that at least a subset of these fold. Now, we're still in the realm of design problems that can be solved on classical computers, but these are some of the largest design problems that we can solve on classical computers. And with improvements to the hardware, we expect that very near generation quantum computers will show advantages over classical computers. When that happens, we're ready for it. So with that, I want to thank all of you for your attention, and I want to acknowledge the people who contributed to this work, particularly Doug Renfrew, who co-heads the biomolecular design group with me, Varjin Turzo and Xiao Zhu, who are excellent postdocs with brilliant ideas, whom I highly recommend. Noura Azadvari, who is a graduate student who worked with us on Masala, very capable graduate student who will be graduating before too long. Tristan Zaborniak, who was a graduate student and recently defended his PhD and is looking for postdocs, who has worked on the quantum computing stuff and is absolutely brilliant. Highly recommend him as well. Our collaborators at UCLA and Vanderbilt and in industry. And as a final note, if you're interested in our software, Rosetta is free for academic use and is licensed to companies for a fee. Masala is free and open source for anything you want to use it for. We have a preprint on bioRxiv. We have a methods in enzymology paper out now. And here are all the repositories for Masala, so you can download the code yourself, extend it yourself, do what you like with it yourself. If you want details on how to use these pipelines, these 3 book chapters provide very detailed information about them, or we're always looking for collaborators, and we love collaborating with experimentalists because it gives us an opportunity to improve our methods with real experimental feedback, and hopefully it's fruitful for the experimentalists as well. Thank you kindly.